Hi, everyone. So Naveen Nagarajan is a faculty of the Department of Pediatrics Research Institute at the University of Louisville. Before joining U of L, Naveen worked with Dr. Mario Capecci at the Department of Human Genetics at the University of Utah. His work is focused on investigating the neural circuit and cellular mechanisms underlying microglia neuronal interaction and repetitive behaviors using multidisciplinary neuroscience approaches that include genetics, behavioral, optogenetics, miniature fluorescence endoscopy, electrophysiological, and computational approaches. Dr. Nagarajan received his Ph.D. in chemistry with a specialization in biophysics and neuroscience from the Department of Membrane Biophysics, Max Planck Institute for Biophysiological Chemistry in Göttingen, Germany, under the guidance of Dr. Christian Rosamund and Dr. Ewan Neher. He did a postdoctoral fellowship in cellular neuroscience in Mark Baer's lab at the Piquar Institute for Learning and Memory at MIT, and systems neuroscience at the Keck Center for Integrative Neuroscience at the University of California, San Francisco, with Dr. Michael Mezenich. He joined Dr. Mario Capecci's laboratory at the Eccles Institute of Human Genetics at the School of Medicine, University of Utah, as a postdoctoral fellow to investigate the role of HOXB8 gene function in repetitive anxiety and social behavioral functions and the HOXB8 mutant mouse model for repetitive behaviors. Using optogenetics and miniature endofluorescence imaging technologies, the team for the first time discovered that the optogenetic stimulation of HOXB8 microglia could induce repetitive grooming behavior in a control mouse. This resulted in a paradigm shift in how the brain utilizes unique microglial functions, capabilities to generate specific behaviors in a circuit-dependent and cell-specific fashion. These studies provided a novel research direction and insights to unravel the mechanisms of microglia neuronal communication and repetitive behaviors. The communicative signals between the brain's immune cells and the neuronal circuit is the first of its kind and will lead to the elucidation of how mutations in immune cells, the response of the neural circuit, and the brain microenvironment directly impact the cognitive, emotional, and social function of the brain in healthy and autism-specific disease states.
Thank you very much for your kind introduction, and good morning to everyone. Thank you for joining my talk. So today what I'm going to do is to tell you about a subpopulation of microglia, which was discovered in Mario's lab. And this subpopulation of microglia is labeled with a specific gene, HOXB8 gene. And I hope by the end of the talk, you will appreciate, what's the role of this particular gene and how genetic mutation in this particular gene leads to a very specific behavioral output, that is repetitive behavior, repetitive grooming behavior, and anxiety like behavior in mouse model. And you would also appreciate, the localization of this particular gene in a specific cell type in the brain, and that turns out to be microglia. So why are we interested in this particular gene and this particular behavior? Repetitive behavior is a common behavioral output that is observed in many neuropsychiatric conditions and neurodevelopmental disorder. Most prominently, it is observed in autism as well as OCD-like behavior. And if you look at the repetitive behavior, the behavior itself is an evolutionary conserved behavior. It's found in invertebrates like Aplysia, C. elegans. It's found in Drosophila, zebrafish, and goes to rodents like mouse and rats, and also found in non-human primates. And it extends even up to humans. And repetitive behaviors during early development could be manifested as hand flipping behavior, which is a common behavioral output in autistic children, or repetitive lining of objects, which many of you might have found in autism spectrum disorder. And because they are locked in this repetitive behavioral chain of behaviors, they also display a lot of speech regression, which is caused by not being able to communicate the information. And that further leads to social isolation as they grow older. So if you look at the repetitive behaviors in adults, it's detected as mental obsessions. And approximately 1.2% of the population suffers from one or the other type of obsessive compulsive disorder, which is actually caused by repetitive thoughts. And we know that fear or anxiety, like stimuli, can enhance this obsessive compulsive behaviors. And approximately 6.8 million people suffer from repetitive behavior, as well as fear or anxiety induced repetitive behavior. And in worst cases, it also leads to panic attacks. And this has escalated post-COVID-19. So if you look at the symptoms and comorbid features of the repetitive behaviors, we can classify them into genetic comorbidity, cognitive comorbidity, behavioral comorbidity, as well as medical comorbidities. And several genes have been found to be associated with autism spectrum disorder, and those are shown in these panels. And if you look at the cognitive comorbidities, you see that they display intellectual disability as well as language impairment. And behavior-wise, they display impulsivity or hyperactivity, agitation, and anxiety like behavioral states. And medical comorbidities includes severe constipation, as well as in low functional autistics, the kids also display seizures, epileptic seizures. So several biomarkers have been tried and used to identify if there are any way that one could attempt to detect the autism early during the development. And some of those attempts are shown here. This includes altered immune or mitochondrial indices, or fMRI imaging, or abnormal EEGs. So all these biomarkers are now being used to at least say that there is something that's going on wrong during the early development. If you look at the symptoms and comorbid features of the repetitive behaviors pertaining to OCD, you see that OCD people, they manifest the OCD like behaviors through body dysmorphic disorder, Tourette syndrome, chronic tic disorder, as well as eating disorder. And in several cases, they also display what is called trichotillomania and grooming related disorder. And this is a particular disorder that I'll be focusing for the next few minutes. So if you look at what are the approved drugs for autism, it turns out to be only two drugs, risperidone as well as aripiprazole. Both these drugs are non-specific drugs, and they are not going to cure autism, but they are going to suppress some of the symptoms associated with the core autistic behaviors. And risperidone is again a common neuropsychiatric drug that's given for psychosis or depression and bipolar disorder. So it's not a specific drug. However, if you look at all the factors that contributes to ASD-like behavior, we see that there are genetic and non-genetic as well as epigenetic component on one hand, but also several of the other factors such as environmental toxicants, maternal immune activation, microbiota, metabolic pathways, neuromodulation, as well as maternal stress that could be important for inducing the early developmental defects associated with autism in newborns. So as a parental responsibility, a parent who are having ASD kids, they have responsibility of not only the medical care as well as education concerning the child they have, but they also have the responsibility of thinking about what's the future of the adult autistics. How are we going to deal with the careers or jobs, or how are we going to deal lifelong comorbid conditions that are associated with autism? and also, several of the safety concerns concerned with the regular safety associated with the autism management. And more challengingly, it also drain on the parents as to how they self-manage, as well as how family need to manage, the autistic individuals as autistics grow older. So there is a very important reason why this research is important, not only for from the scientific standpoint, but also from the standpoint of the child management in early development. So in this particular journey, my interest in this repetitive behavior was to find out a simplest form of this behavioral output um, where I can associate genetic function to the behavior. So in my search, I found that there was a specific gene called HOXB8 gene that was discovered in 2002. And genetic mutation in this particular gene, which leads to loss of function of this gene, leads to excessive grooming behavior. And by 2010, in Mario's lab, it was already found that the gene was present only in microglia in the brain. And especially 30 to 40% of the entire brain microglia contained HOXB8 gene. And when I joined the lab, what was unknown was what was the neural circuit on which these microglia were acting in order to induce this behavioral output. So why do we have to care about neural circuit in relation to repetitive behavior? Neural circuit, we all understand, process information via fundamental units called synapses. And synapses are the fundamental unit of the brain. And my, reductionistic approach was to connect a particular neural circuit to the behavior through HOXB8 gene or HOXB8 expressing microglia. And that's the chain of information that I was trying to understand in this project. So even before we attempt to understand everything, what we need to understand is what are the known functions of microglia and what do we know specifically about HOXB8 microglia. So I don't have to mention to this audience or any audience, how important microglia are for neurodevelopmental, neuropsychiatric, as well as neurodegenerative diseases. Open any of the top journals or any of the work that has been accomplished in many of the brain diseases, you would see microglia to be playing very important role, be it depression, be it multiple sclerosis, addiction, or Alzheimer's disease. So there is a very important role of microglia as well as microglia contribution to the onset as well as the sustainment of the disease. And my interest was to understand what was going on with the HOXB8 microglia in the brain. So what we knew earlier that was that HOXB8 cell lineage gives rise to brain microglia. So how do we do this? You take a HOXB8 Cre driver, Cre is a enzyme, and then you cross that mouse with a female mouse that contains a fluorescence reporter like Rosa YFP, so that you can express all the fluorescent protein YFP only very specific to the HOXB8 cells. And if you do that cross, and if you look at the pup's brain, all what you see is microglia that are shown here. And how do we know that these are microglia? You can use a very specific marker like CD11B, which is a pan microglial marker, and you see the co-localization that is shown in yellow. But what you would also appreciate is that there are several cells that are not marked with, the HOXB8. So that suggests that these are non-HOXB8 microglia. And if you look at the early development from postnatal day 2 all the way up to postnatal day 90, you see that there is emergence as well as infiltration of HOXB8 microglia slowly from postnatal day, and it infiltrates by P14, and by around 1 month, you get complete maturation of these microglia and it fills the brain. We discovered later that these microglia are coming from bone marrow. And now we understand that even before bone marrow, HOXB8 microglial precursors are found in the fetal liver. So traditionally, it was assumed that all microglial precursors come from yolk sac by around embryonic day 9.5. And our lab identified that that's true to some extent, but HOXB8 is very exclusively expressed between embryonic day 12.5 and 14.5. They use a second wave of hematopoiesis within the fetal liver. So the origin is from a different location, and the time of entry of these HOXB8 microglia in the brain is also different compared to non-HOXB8 microglia. So since I mentioned that the precursors come from bone marrow, a bone marrow transplantation experiment was conducted, and in this experiment, it was transplanting the healthy bone marrow from a healthy mouse onto the mutant mouse and reversing the same experiment by taking the bone marrow from the diseased mouse and putting back into healthy mouse. So what they found was that by transplanting the healthy HOXB8 microglial precursors, the CD34 positive cells, in bone marrow, they were able to completely rescue the behavior as shown here. And this was observed in 60% of the animals that received the bone marrow. And quantification showed that the mutant animals spent twice the amount of time in grooming. However, the rescued mutant show wild-type-like behavior. And if you introduce the unhealthy disease HOXB8 microglial precursors back to a healthy animal, the healthy animal will now start developing the disease. So in either case, the experiment succeeded, and it proved that there is something that is important in relation to what happened post bone marrow transplantation, which led to the rescue of the behaviors. And one thing that was not clarified in this paper was what happened, inside the brain. What precursors or what microglia, or whether microglia and macrophages rescued the behavior, or what's the direct correlation between microglia and the behavior. So now that's where I asked a simple question as to what's the role of HOXB8 microglia in the brain that has led to the induction of these behaviors. And in order to address that, first we have to understand something about HOXB8 microglia and non-HOXB8 microglia. So the question is, how are they different from each other? So in order to study this, you have to look into the distribution of these microglia in the brain. You have to study what are their morphological features. How do they dynamically sense their projections while the brain is developing? How do they interact with neurons and synapses? And what are their physiological properties? And finally, if you create any damage to the brain, how are they responding to that particular damage? So if you look at the distribution of HOXB8 microglia in the brain, shown here is a sagittal section of the brain, where we have labeled all microglia in green color with IBA1, which is a microglia marker. And on the top of it, the red microglia corresponds to the HOXB8 microglia. And what you see is the unequal distribution of the HOXB8 microglia. For instance, in orbitofrontal cortex, it's 40%. In motor cortex, it's 61%. If you look at areas like VP or substantia nigra, there is reduction of the HOXB8 microglia in these locations. So unlike non-HOXB8 microglia, which is uniformly distributed, HOXB8 microglia is patchy, and HOXB8 microglia is distributed non-uniformly. And that might also have some implication as to why that is happening in the brain. So if we look at the morphological features, HOXB8 microglia exhibit enormous complex projections during postnatal maturation, as shown here in the bottom panel, where you see, quite number of primary filaments and secondary filament and tertiary filaments, while non-HOXB8 microglia exhibit more reduced number of projections. And this increase in the number of projections also decreases as the brain matures. And this was later on studied using two-photon in vivo imaging as to why they need to have that many projections and what's the role of these projections and how are they dynamically interacting with the neighboring synapses or neighboring neurons. Shown here is a P32 brain that 1.5 millimeter down inside the brain, showing you the distribution of HOXB8 and non-HOXB8 microglia. And here are the projection dynamics of non-HOXB8 microglia and the HOXB8 microglia. And when you quantify, what you see here is that during critical period, which is between postnatal day 8 and 16, or during pre-critical period, which is postnatal day 0 to 8, you see that the dynamics of non-HOXB8 microglia is very distinct compared to HOXB8 microglia. HOXB8 microglia over time, gives more projections and retracts the projections more with increased length as well as with increased amount of the speed compared to non-HOXB8 microglia. And that's what is shown in the heat maps here. So you see that during pre-critical period or critical period, the activity of the HOXB8 microglia is way more higher compared to non-HOXB8 microglia, and that will wane down once we cross the critical period. So obviously, microglia are interacting with the neurons and synapses. And if you look at the non-HOXB8 population and HOXB8 population as to how they are interacting with the neuronal surfaces, what you see here is there are several puncta that is on the HOXB8 microglia that one could detect more readily as compared to non-HOXB8 microglia. And all these puncta are coming from the neuronal surfaces that are touching the HOXB8 microglia. And that suggested that HOXB8 microglia might be interacting with the neuronal surfaces more readily compared to non-HOXB8 microglia. And to prove that in much more rigorous fashion, I looked into Ph.D. 95, which is a marker for dendritic spines, as well as which is a postsynaptic density protein, and noted that HOXB8 microglia projections exhibit enormous number of punctate staining on the projections compared to non-HOXB8 microglia. That also suggested that the co-localization of HOXB8 microglia with the Ph.D. 95 is much more stronger compared to non-HOXB8 microglia. How are they different in terms of their physiological property? A simple way of addressing that is using electrophysiology. So you can patch individual microglia, green as well as red microglia, and characterize very basic properties such as membrane potential, capacitance, and resistance. And I noted that none of these properties were different in HOXB8 versus non-HOXB8 microglia, and that's what is quantified here in terms of the currents elicited across various holding membrane potential, and no significance. However, when I looked into the response to ATP, what I found was that exposure of 1 millimolar of ATP to HOXB8 microglia resulted in reduction of the current amplitude compared to non-HOXB8 microglia. And that was significant both at minus 70 millivolt as well as at minus 30 millivolt. Likewise, if we look at the current amplitudes at the positive membrane potential, we still saw a trend in the decrease in the current amplitude. However, this was not significant. So at least we learned that their response to ATP is different compared to non-HOXB8 microglia. The next question was, do they respond similar or differently to any brain injury? And in order to address that, I looked into the two-photon in vivo imaging, where I ablated a small location inside the motor cortex. And the experiment is to shoot the two-photon laser for about 1 minute in the motor cortex and watch what happens to HOXB8 microglia and non-HOXB8 microglia. And this is traditionally used in the field to find out whether microglia are normally behaving or do they have any abnormality in response to normal condition or in response to disease or in response to any kind of damage. So in conducting that experiment, what we noted was that HOXB8 microglia sends more bulbous projections to the location of the damage that is induced here compared to non-HOXB8 microglia. Secondly, HOXB8 microglia also collaborates with the other microglia in order to send their bulbous projections. And they are sending their bulbous projections to remove the apoptotic signals or apoptotic materials that are produced in response to the damage that I created by the lesion here. So there were similarities, but there are also differences. So next, the question is, how do HOXB8 microglia communicate with synapses and the neural circuit? So in order to address that, we don't even know what neural circuits are playing an important role in the brain and on what neural circuit HOXB8 microglia is working to induce these behaviors. So it was quite a black box for me. So to address that very systematically, we have to go about in first learning what connectivity are changing in the brain. And once we detect that there is a neural circuit that is defective or that is altered in the HOXB8 mutants, then we can go very deeply into that particular circuit to identify what's going on at the neuronal level as well as at the synapses level. So to study that, the first experiment was to do awake fMRI imaging for about 60 minutes and look at what's going on in the brain of the healthy mouse as well as the HOXB8 mutant mouse. So shown here is the awake fMRI chamber in which the mouse is allowed to sit for 3 days, during which the conditioning happens. Once the animal sits without shaking the head or without any disturbance, then based on the learning of the ability of the mouse to remain still, we can measure the fMRI signals. And shown here are the functional connectivity from 21 brain regions demonstrating that the connectivity of the right hippocampus to the left hippocampus or right caudate putamen to left caudate putamen on either of the hemisphere are equally homogeneous, and these connectivity are all intact in a wild type animal. So if we perform the same kind of connectivity analysis in the mutants and compare to wild type, what we discovered was that a lot of connections, which was supposed to fall on this correlative line, it turned out that a lot of these connections were hypoconnected. They were all hypoconnected in the knockout compared to wild type. So then we questioned, is this the common phenomenon that's going on in the entire brain, or are there any regions inside the brain that might show altered connectivity other than hypoconnectivity? So to address that, we used seed analysis of creating seed between striatum and the cortex and map every connection between striatum and cortex and started looking into these corticostriatal connections in the wild type and the knockout. And shown here are those connectivity maps where we detected that the signals in specific regions of the brain, especially the connections of the striatum to the frontal cortex, was hyper in the mutant compared to wild type. And that's what is shown in the maps here, wherever you see the hot signals like red or, those are the signals where the connectivity is way more higher in the knockout compared to wild type. And wherever you see the dim signals, those are the connectivity that are hypo or the connections that are less connected. So in this analysis, we detected that the connection between medial prefrontal cortex and striatum, as well as the orbitofrontal cortex and striatum, was hyperconnected, while all the other connections were hypoconnected. And if you look at the female samples compared to the male samples, we see that in females, HOXB8 mutants display way more hyperconnectedness compared to the male samples. And that gave us an indication that, yes, now we have to focus on corticostriatal circuitry. And from here, what do we do? So we can go to individual neurons, individual synapses, and see what's going on in frontal cortex as well as the striatum synapses. So I use two technologies. One is electron microscopy and here, and then the Golgi Cox staining here to determine what is going on with the dendritic spines as well as what is going on with the synapses. So by looking at the dendritic spines in the frontal cortex as well as striatum and analyzing that data set, what I found was in the knockout, the number of dendritic spines of the cortical neurons was way more significantly higher compared to wild type. However, if you look at the striatal neurons, we found opposite effect that the dendritic spines were significantly reduced in the knockout compared to wild type. And if you look at the electron microscopic images of the cortex as well as striatum and start measuring the length of these synapses, we found that in cortex, the asymmetric as well as symmetric synapses, which corresponds to excitatory and inhibitory synapses in the brain, they were all showing increased synapse length, which was significant compared to the wild type. However, if you look at the striatal synapses, we found opposite thing that the synapses were contracting. So that was what is modeled here. Shown here is the connectivity that I talked just now. There is hyperconnectivity between very specific brain region, medial prefrontal cortex, dorsomedial striatum, but all the non-corticostriatal connections were hypoconnected. And here is the summary slide, which shows that the synapses are expanding in the cortex, while in striatum, the synapses are contracting. And that indicated that there is structural defect associated with the synapses within the corticostriatal circuit. And obviously, if there are changes in the density and if there are changes in the synaptic structure, one would ask what's going on with the synaptic plasticity in the brain. And to address that, I did two experiments. One is short-term potentiation experiment, and second one is long-term potentiation experiment. And these are the standard synaptic plasticity experiment where you conduct electrophysiology and you electrically stimulate one part of the brain and record the signals from the neurons that are connected to that part of the brain. And in this particular case, I looked into corticostriatal synapses. The experiment is simple. It's not simple, it's complex. It's simple to explain, which is, you put a electrode, stimulating electrode in the deeper layer of the cortex, and then you record with a recording pipette inside the striatum. And by doing that, and by giving a paired pulse, I found that the amplitude did not change significantly in this paradigm, which is called paired pulse paradigm. And that, if there are changes, then we say that there are changes in short-term potentiation. But since there are no changes, there was no change associated with short-term potentiation. However, when we looked at LTP, which is a much more long-term potentiation, the experiment is, again, giving high-frequency stimulation to the stimulating electrode on the presynaptic neuron, and then measuring what's going on with the electrical amplitude in the postsynapse neuron. What we found was that over time, the knockout showed exaggerated response compared to the wild type. And this was true for a specific type of LTP called CNQX, insensitive form of LTP. And the amplitudes were collected from this response called S1 response, which is a presynaptic component, while the postsynaptic component, which is corresponding to S2, that did not show any significant difference. So this suggested that it is presynaptic LTP that is impaired compared to postsynaptic LTP, which means the defect is associated with the projections that are coming from the frontal cortex and reaching to the striatum. So besides the grooming behavior, I also looked into anxiety behaviors and found that these animals exhibit anxiety-like behavioral states where the animal remain in the closed chamber and they do not enter into open chamber as compared to the wild type animals. And likewise, if we do a open field test, the animals spend more time in the periphery as compared to spending the time at the center. So both the center zone as well as the open zones are the anxious inducing zones or anxiety inducing zones, which is what is being avoided by the HOXB8 mutants compared to wild type. And I showed that, quantification wise, that these animals spend less time at the center as well as less time in the light zone, meaning that these anxiety levels in these mutants are way more higher. So in a three-chamber assay, I also showed that the knockout mouse does not interact with an intruder mouse more readily as compared to the wild-type mouse. In the wild-type mouse, when you expose to an intruder mouse, it readily interacts, but HOXB8 mutant mouse does not interact. And that's what is quantified here. So that suggested that even social behaviors is impaired. So next, I looked into some pharmacological treatment. And since fluoxetine is one of the common SSRIs that are given for many of the autistic kids as well as OCD patients, fluoxetine treatment seemed quite appropriate for this mouse model. By administering 5 mg per kg fluoxetine for 5 weeks, I was able to find that the grooming behavior can be rescued without affecting the non-grooming behavior such as locomotion or the immobility. Likewise, if you look at the anxiety behaviors, either using plus maze, open field, or light dot test, you see that one could rescue the anxiety in response to the treatment conditions of the fluoxetine. Social behaviors improved with the fluoxetine treatment. So the animal that did not interact, the HOXB8 mutant, now started interacting more readily with the intruder in response to fluoxetine treatment, which suggested that social behavior is also improved with this treatment condition. So, so far I showed you that the grooming behavior, anxiety behavior, and social behaviors are impaired, and one could pharmacologically rescue these behaviors. And I also showed you what circuit was playing an essential role and how dendritic spines or synaptic structures are altered and plasticity is, affected in this particular circuit. So now the big question was, how can we associate all the information that I have shown you to the microglial deficits in the brain? And in order to study that, you have to go deep into microglia to find out what is going on inside microglia. And to start with, I'm just showing you here the distribution of the microglia in these regions, the frontal cortex, striatum, cingulate cortex, and thalamus. And like I mentioned earlier, the distribution is non-uniform in these brain regions. Again, red corresponds to HOXB8 and green corresponds to non-HOXB8. And if we do the same experiment of brain slices and look into the frontal cortex and striatum and quantify what's going on in the mutant mice and compare that to what's going on with the wild type mice, I found that the mutants display reduced density of HOXB8 microglia as well as non-HOXB8 microglia compared to wild type. The same was true for the dorsal striatum as well. So that suggested that the density of microglia might be one factor that is affected by HOXB8 mutation. What about morphology? So if you look at the morphological features of the knockout and compare that to wild type, and if you look at what's going on in the mutant compared to wild type, what you see here is that the HOXB8 microglia shows reduced morphological projections compared to the HOXB8 microglia in wild type condition. And that is true for both medial prefrontal cortex and dorsomedial striatum. And that's what is quantified here, that the primary projections, secondary projections, or tertiary projections are all significantly reduced in the knockout compared to wild type mice. So there is defect in the number of projections that they have. Density is number one, morphological feature is number two. So one could ask the same question as to what happens if you induce 2-photon induced laser injury in the mutant mice. And surprisingly, what I found was that the projections did not come to the location where the damage was induced. So that suggested that although the microglia with the HOXB8 mutation exhibit the projections are not capable of going to the damaged area. So that also suggests that they fail to respond to the laser-induced injury. So for less than a minute, I want to show you what is exactly going on inside the HOXB8 mutant brain without going inside the brain, but just imaging inside the brain. And this is a 2-photon in vivo imaging again, showing you a live mouse breathing and under anesthesia with its with a craniotomy and with a 2-photon in vivo imaging the brain for about 600 to 700 micron deep inside the frontal cortex. And what surprisingly came out of this experiment was that you can see that there are several locations inside the brain where microglia is not even entering, and there are several locations where microglia is accumulating. So this is not a normal distribution inside the brain. So if you do a control experiment with the same kind of two-color mice where we have green and red microglia, what you see here is a complete uniform distribution of microglia everywhere. But here we found that there is a hole inside the brain. And that is also very important to know what is the meaning of that hole, why microglia are not coming into this location. And if you remember the hyperconnectivity and hypoconnectivity that I was mentioning, one reason why microglia might not be coming here is because there is something that is released in that location which is repelling microglia, so microglia are not entering into this zone. We don't know what that molecule is, but that is a speculation hypothetically. So, but let's imagine what happens if microglia doesn't come to that location. That means, all those connections that need to be pruned are not getting pruned properly. Okay. And what is the meaning of all microglia going and accumulating other brain regions? What it means is wherever there is no necessity of additional microglia, if microglia go there, microglia just start pruning a lot of synapses. And that could be explaining why we are seeing hypoconnectivity in some regions and very specific hyperconnectivity. So hyperconnectivity could be that the synapses not being pruned in these frontal cortical, striatal connections, but hypoconnectivity may mean that a lot of synapses have already been pruned more than what it need to be pruned, and that's why you are seeing hypoconnectivity. So to address that in more detail, I also looked into microglia neural interaction and found that with the reduced number of projections, HOXB8 microglia is touching way more number of neurons than compared to the wild type microglia, as well as the non-HOXB8 microglia. So they have reduced projections, but they touch too many synapses. Okay. And that's what is quantified in here. Obviously, a reviewer's question is, what's going on with the cytokine levels? Because microglia, we know that releases cytokines, and or do we see any differences in cytokine levels? So I looked the cytokine levels in mutants and compared that to wild type, and noted that the pro-inflammatory cytokines like Kc or IL-17 showed an increase. However, the anti-inflammatory cytokines like IL-4, showed a significant decrease. And these were the cytokines that were detectable in our samples. And I'm not showing you the entire panel of the cytokines that we did, but at least these indicated that there are significant changes associated with the pro as well as anti-inflammatory. And that could also point to the mechanism as to what might be happening within the microglia and what microglia might be releasing in response to the genetic mutation. So this was the model that was proposed in 2010, that there could be a circuit like this, like corticostriatal circuit, that might be defective. And I showed you that cytokines up till now like IL-17, IL-4, TNF, alpha, and KC might be playing essential role in imbalancing the cytokine levels in order to induce these behaviors. So in the last part of my talk, I want to go into very causality level question. So in most of the papers that I published or we submitted, most important question was, what is the causal link between HOXB8 microglia and the neural circuit as well as the behavior? So that causality link question is a very complicated question, because what they're asking is to prove that is there any direct role of HOXB8 microglia in the brain to the behavior, not with genetic mutation or anything like that, but the actual role of HOXB8 microglia to the behavior. And that can be addressed using some elegant neuroscience technologies like optogenetics and miniscope imaging. And the last several years, I spent in establishing and developing these techniques applicable to microglia, and you'll see, what I came up with. So to conduct optogenetics, the simple question is, can we do optogenetics on microglia? And if you are doing optogenetics on microglia, HOXB8 microglia, what circuit do we need to focus? And I focused on corticostriatal circuit and anxiety circuit because that was more relevant to the behavioral phenotype. And I looked into the same regions that we saw hyperconnectivity, the dorsomedial striatum, medial prefrontal cortex, as well as anxiety circuits where basal lateral amygdala, central amygdala, and the ventral CA1 region of the hippocampus. So the experiment is to express this AI32, which is channel rhodopsin, a light-sensitive protein, inside HOXB8 microglia, very specifically using Krelok's technology, and then shine light on these microglia and ask a simple question: do we see any change in the behavior in response to the light stimulation? So the first question is, can we express channel rhodopsin in HOXB8 microglia? And can we be specific to that? So here are brain slices showing you that we can make channel rhodopsin very specific for HOXB8 microglia. So all these green microglia that you are seeing, right now, they're all HOXB8 microglia, and they're labeled with IPA1 to show you that these are subpopulation of microglia that are labeled with IBA1, but there are also other microglia, non-HOXB8 microglia, that are not expressing channel rhodopsin. Okay. So they don't co-localize with neurons, as shown here with the new N marker demonstrating the specificity of channel rhodopsin to HOXB8 microglia based on the Krelok's technology. So the experiment is, you put a cannula in this location, dorsomedial striatum, at this stereotactic coordinate, and directly shine light on the striatal HOXB8 microglia. And in response to light, I was able to demonstrate that HOXB8 AI32 mice displayed significantly increased amount of grooming time, as well as increased number of grooming bouts compared to C57 black 6 mice. So if you quantify what's going on with the pre, during, as well as post-stimulation condition, you see that in C57 black 6 mice, there is no significant difference. However, if we look at the during stimulation compared to pre and post, for the HOXB8 AI32, we see there is a significant increase in the grooming behavior as measured by grooming time or grooming bouts. Here is a video of an animal that's undergoing optogenetic stimulation. So that's the pre-stimulation condition, normal behavior. You don't see any grooming or anything like that. And here comes the light stimulation. It takes about 20 to 30 seconds for the initiation of the grooming behavior. And just to mention that these are all control mice. They don't have any genetic mutation or anything like that. But you start seeing that upon light stimulation, the animal starts grooming. And this is a very typical facial grooming induced by striatal microglia, HOXB8 microglia stimulation. And after the facial behavior, they also display the body grooming, which is a phase 4 type of grooming. And it keeps on going on and on and on as long as the light stimulation is going on. And once you come out of the light stimulation, the behavior stops. So that suggested that HOXB8 microglia stimulation is indeed able to induce the grooming behavior. Now, if you look more carefully inside the brain where we stimulated, you see the HOXB8 microglia, and these microglia are not in activated state, but they show the same morphological features, demonstrating that we are not inducing any disease-like states by stimulating HOXB8 microglia, nor do we see any excessive grooming behavior once the light is turned off. So a healthy mouse just starts showing the grooming behavior upon shining the light, but it shuts down once the light is off. So, if you repeat the same experiment in medial prefrontal cortex, you observe that you see the same increase in the grooming time percentage as well as grooming number of bouts. And similar analysis led us to conclude that medial prefrontal cortex HOXB8 microglia also leads to the grooming behavior. So shown here are histological slides, for the verification of where I implanted the cannula, and the microglia are all intact in these animals. And you can do these experiments on and on and on several times. You can reproduce the experiments, you can do multiple sessions of the experiment, but these are all, the behaviors come back when light stimulation is given. So I then also looked into what happens if you induce optogenetic stimulation of HOXB8 microglia in amygdala, because this is a whole different brain region that controls fear and anxiety, like behavioral states. And the question was, does stimulation of HOXB8 microglia just induces grooming behavior, or is it capable of inducing anxiety-like behavioral state? And both in basolateral amygdala and central amygdala, I was able to show that you can induce the anxiety-like behavioral states where the animal remains in the closed chamber and never enters into the open chamber, avoiding the anxiety-like behavioral state, and that's what is quantified here. So shown here is the implantation site and the histological verification. And I also showed that a second region, ventral CA1 region of the hippocampus, when stimulated, also show similar kind of anxiety-like behavioral states in HOXB8 AI32 animal. But in C57 black 6 mice, you don't see that behavioral output. So you can conduct an open field test and observe the same kind of anxiety-like behavioral state where the animal tends to remain in the periphery and do not enter into center brain region. However, a C57 black 6 mice stimulated by giving the similar kind of stimulation within the same region, they show normal distribution of activity in the entire open field. So interestingly, by stimulating ventral CA1 region of the hippocampus, I also noted that it not only gives rise to anxiety-like behavioral state, which then turns out to freeze the animal, but we can also induce the grooming behavior, and that's what is quantified here. Both in the home cage behaviors, both the percentage of grooming as well as percentage of freezing was significantly higher in the stimulated condition compared to the non-stimulated condition or the C57 black 6 mice. So some more verification of the ventral CA1 region of the hippocampus. One question was, what happens if you stimulate all microglia inside the brain within this particular brain region? And to our surprise, what we found was when we stimulated all HOXB8 and non-HOXB8 microglia using IBA1 Cree driver, surprisingly, the behavior shut down. There was no behavior whatsoever. And it was very specific for only HOXB8. And this was proved in multiple ways and multiple brain regions. And anytime anybody try to stimulate all microglia, behavior shuts down. Okay. And I showed electrophysiologically that, not only the continuous blue light stimulation induces an increase in the neural activity in response to light stimulation, but even 80 hertz high frequency stimulation can increase the spike activity, in the patched neurons. So quantification is shown here. So interestingly, in response to light stimulation, CFOS, which is the early gene expression marker in the neuron, they get turned on. However, if you look at IBA1 mice, the CFOS is not increasing, which suggests that something is not going on, something is preventing the CFOS expression in the neurons in response to all microglia stimulation compared to HOXB8 microglia stimulation. And likewise, when I looked into HOXB8 microglia alone, to what happens after the light stimulation, I was able to find that we can increase the Kv1.3 expression, which is a potassium channel marker, suggesting that the membrane is being depolarized in response to light stimulation of HOXB8 microglia. So this is a model we came up with to explain. And here is an accelerator and brake model. We think that HOXB8 serves as a brake in the brain, and non-HOXB8 microglia serves as an accelerator. Oops. And by creating a genetic mutation or by inducing optogenetic stimulation, we perturb the brake. And by perturbing that brake, we induce a repetitive grooming behavior or anxiety-like behavioral states. And that dynamics of accelerator and brake model probably is intact in healthy animals. However, the imbalance between the brake and accelerator might what leads to the grooming behavior and anxiety-like behavior. And in last couple slides, I just want to show you what's going on with the physiological role of HOXB8 microglia inside the brain. And in this experiment, the question is, in response to the shift of the behavior from normal behavior to grooming behavior, can we detect something inside the microglia? Can we have a physiological signature of the microglia? And you can do that by putting a calcium sensor like GCAM5 and very specifically expressing GCAM5 inside HOXB8 microglia as shown here. And the experiment is simple. You spray a little bit of water on a healthy animal, the animal starts grooming. And in response to grooming, you put a mini scope as shown here on the brain and see what happens to those microglia. And what I found was you can in fact detect increase in the calcium signal in these individual microglia in response to the behavior stimulation that sustains for as long as the behavior is present. But once the behavior shuts down, again, the calcium goes down. So it's almost like a tuning of calcium in alignment with what's going on with the behavior. So every time the behavior changes, you can also see a change in the calcium levels. And these are the calcium transients, and these are real calcium transient, because if you put a ROI on a region which is not a cell, you get oscillatory activity, but you don't get real calcium transients. So shown here is a correlation plot where you see, the pictures of pre-stimulation condition. There is no signal of calcium increase in microglia. And in response to behavioral stimulation, you see a lot of calcium increase. And then post-stimulation, you don't see that. Okay. And here are the compiled heat maps of all the microglia, pre-stimulation, during stimulation, and post-stimulation condition. And in conducting the correlation plot, what we found was that activity of all the microglia is highly correlated, because the calcium levels in all microglia was going on simultaneously up in response to the behavior stimulation. So it's not, one microglia calcium is going up, which is then triggering the next microglia, but rather everything is going in concert with each other. And that's the reason why we see that the activity is highly correlated. So if you repeat the same experiment in medial prefrontal cortex, you end up in getting the same type of calcium transient in response to behavioral stimulation. And you can, based on the number of grooming bouts that is, that the animal is exhibiting, you can also get as many calcium transients. So if you have multiple bouts, you can get multiple calcium transients. And if the animal sustains a single grooming bout for a longer time, you can also induce a huge calcium transients. So if you sustain the grooming behavior for a longer time period, you can get multiple calcium transient. And this I did by using a small oil drop on the surface of the skin of the mouse, which resulted in mouse grooming excessively. And in response to that excessive grooming, I was able to capture calcium transient, in all the microglia. You can classify microglia based on the shape of the calcium transient, but you can also classify microglia based on the activity itself of individual microglia. And here are the histological verification that these are the microglia that I saw inside the lens, inside the brain, while I was measuring the calcium signals. And when we asked the question, what is the nature of the calcium transient in HOXB8 mutant, to our surprise, what we found was we couldn't detect any calcium change in HOXB8 mutant microglia. All the lines were pretty flat, and we could not detect any calcium regulation. So to explain what I'm trying to say, when you image the GCAM signal inside HOXB8 mutant microglia, the calcium level remains turned on all the time. The calcium never comes down. If you look at a healthy mouse or a healthy microglia, calcium goes up and comes down, because of which we see calcium transient. But here, it doesn't happen. So that could be a mechanistic link, and that was time 0 and time 30. It's the same output, and even if you come, 2 days later, 3 days later, or 30 days later, this is not going to change. So had this been neuron, probably the animal would have died because of high calcium. But because this is not neuron, this is microglia, somehow, brain has been able to manage these microglia with high calcium, but still is able to keep the animal live as long as a healthy, animal, which is quite surprising. So with that, I come to the summary slide. I showed you that HOXB8 mutant exhibit corticostriatal circuit defect. I showed you that the dendritic spines or postsynaptic or presynaptic structures are altered in HOXB8 mutants. I showed you that a specific form of synaptic plasticity is altered in the HOXB8 mutant, in which we also detect the microglia neural interaction to be defective. Optogenetic activation of healthy microglia inside the brain can lead to grooming behavior. And based on which location of the brain you're triggering the HOXB8 microglia, you can induce that particular behavior. And HOXB8 microglia elicit calcium transient in response to the induction of the grooming behavior. And the mutants show the calcium regulation defects. So I would like to come back to my previous slide as to what challenges are ahead of us, and we are at golden time of studying microglia and its association with specific behavioral output, like repetitive behavior as found in autism. And hopefully with all the other modalities, such as identifying what environmental toxicants, affect, could impede the function of microglia, or could induce activation of microglia and alter microglia neural interaction, we may be in a position to identify many of those factors that has not been intensively studied. And that's the hope of my lab in next few years.